So, good evening, students. Today we will discuss block 5th, experimental embryology. So, in this block, 4 units are there. Already in parthenocarpy, the previous class is parthenocarpy is completed. So, remaining topics is experimental biology, application of embryology in taxonomy, agriculture and horticulture, principles and application of paleontology. Bye-bye. So, today we will discuss experimental embryology. So, experimental embryology, what is the embryology? Already we discussed in the embryology. So, today we discuss experimental. What are the experimentals involved in the embryology? So, here the embryology can classified into three categories. First one is the descriptive embryology, comparative embryology, experimental embryology. So the descriptive embryology is the base for other two hand. It gives as detail about the chronology of developmental events. This complication of the data gives an insight into enormous variation exhibited in the reproduction by plants. It is never complete for any given family as a whole. The comparative embryology. The comparative embryology takes in the inputs from the descriptive and comparison are made among the related families to arrive at truly phylogenetic tree. In other words, the embryology is used as a tool in phylogeny. At the first is incomplete this part, which depends on the first, it is also incomplete. Thus, even currently, there is abundant scope of the both these aspects. So, experimental embryology aiming to manipulate the developmental process by understanding the nutritional and environmental factors that affect reproductive growth. So thereby using a knowledge for the human benefit, be it commercial or scientific, that it is combined approach of the embryology. The plant physiology, genetics, and plant breeding. So to say the least, it is to tailor make the plants the terms in the marketability. According to the scientist Massett, the 19th century was able to induce enlargement of ovaries in some plants by treating them with extracts of pollen and spores. This was pioneering effort artificially inducing growth. Around the same time, the Haberland in the 19th century tried to grow groups in the plant cell and medium of leaf extracts. Through it is unsuccessfully. Next, the scientist White in the 1934 was successful culturing roots of tomato, maintaining of the growth rate, subculturing and nutrient medium for the first time. This was White is called father of plant tissue culture. Who is the father of plant tissue culture? White is the father of plant tissue culture. And later, Gautharet in the 1938 is also established the necessity of the various nutrients in the medium for the growing tissues. It is artificially. Many investigators tried various chemicals, and one such path-breaking discovery of the efficiency of coconut milk as a growth enhancer in the medium by Van Overbeek in the 1941. and Muir in the 1950 developed the nuts culture method, the growing embryos or cells on the endosperms of the other plants. And after that, Skoog and Miller, 1955, working on a plant growth hormones, incorporated them into the medium. So these are only to cite a few examples of contributors to nutrient media. When once the nutrient media got standardized in the many experiments also went on. So most significant of such findings of Polian culture and androgenic haploid production by Panchanan Maheshwari and Guha in the 1966. So here the androgenic haploids by Nitesh in the 1974 and protoplasm fusion by Milchers in the 1978. The future of this line research gene manipulation and rising genetically manipulated in the agricultural produce for the consumers. So the technique can be applied to the control and achieve the fertilization in the incompatible processes, rising pure maternal clones by the nuclear cultures, pure parental culture by the genesis followed by diploidization to overcome seed dormancy by exercising and rearing of embryos and to arise a large population of plantlets to the seedling within the short term. So what are the infrastructure requirements for the tissue to be cultured in the lab has to cater to many criticalities. The tissues are handled carefully. The medium preparation and culture maintenance have to be done under extreme aseptic conditions. So the nutrients offered to the growing cells are also nutrients for the fungal and bacterial pathogens. Overcoming the pathogen contamination is a major problem. So the laboratory should be accommodate provision of very small requirement, keeping view of the needs. The infrastructure has to be planned, elaborated nutrient medium, and the preparation of plant material for culturing are three main steps. Planning has to be done. The first one is the laboratory setup and preparation of explants and media preparation. So here, so it is today we will discuss. Already I told experimental embryology. The experimental embryology branch of embryology. The studies the mechanisms controlling the individual development of animals and plants by means the experiments on living organisms called experimental biology. What are the definition of experimental biology? A branch of embryology, the studies the mechanisms controlling the individual development of the animals, and plants by means of experiments on living organism is called experimental biology. It uses such methods are marking, removal, transplantation, isolation of the body parts and organs. This is the regarding experimental embryology. The first experiments on developing embryo were followed by many others, and So the experimental embryology in contrast to comparative embryology or descriptive embryology, the uses of experiment as a method of investigation. However, the use of experimental method in the itself does not create a science and a branch of science. The new branches of science as created by novel viewpoints and novel problems set before the science. It was the problem, what's the ontogenetic development actually, what is the driving forces behind it, necessity the applications, the experiments after the methods of speculation and pure observation were found the solving the problem. The experimental embryology of plants can be discussed under the following uh, following headings. So the evolution and special modification for adaptations. These are the general of experimental morphology. So here the morphological and anatomical So the morphological anatomical adaptation in the hydrophytes and xerophytes, the stamen and androsium, the development and evolution and dehiscence of anthers, development of the anther from the floral parts, the formation of filament, connective and anther bearing pollen grains, the male gametophyte and microsporogenesis, the development of microspores and its germination to gives rise the male gametes. So carpel and gynosium, the structure and function, evolution and development of formation of ovary and style and stigma, the formation of megaspore. So female gametophytes, the megasporogenesis, development of embryo sac. So pollination and fertilization, transfer of pollen grain to pistil and fertilization, development of the seed and fruit, different types of the seeds and development in the different types of fruits, fruit and seed dispersal. So here the maturation of different types of fruits and dispersal of the seeds. So application of the plant embryology, application of biotechnological principles such as stem, root, and leaf culture, anther culture, so pollen culture, so oval culture, applied plant culture, embryo culture, application of plant genetic engineering. These are the application of plant embryology. These are the main things. The first one is the stem and root and leaf culture, anther culture, pollen culture, oval culture, applied plant culture, embryo culture, application of the plant genetic engineering. So here, uh, culture media micropropagation. Culture media micropropagation. Here, the culture media usually contain the following constants: inorganic nutrients, carbon and energy sources, organic supplements, growth regulators, solidifying agents, and pH of medium. So, first of all, we will discuss So we will discuss in experimental biology. So we will discuss what is the plant tissue culture. The plant tissue culture is a technique of growing plant cells, tissues, organs, seeds, and other plant parts in a sterile environment on a nutrient medium. This is the regarding about the plant tissue culture. So why it is plant tissue culture needed? So the production of clones of the plants that produce particularly good flowers, fruits, and other desirable traits, and to quickly produce the mature plants. The production of multiples of the plants in the absence of the seeds are necessary pollinators to produce the seeds. So the generation of whole plants, sorry, the generation of whole plants from the plant cells have so here to quickly produce the mature plants, the production of multiples of the plants in the absence of the seeds and necessary pollinators to produce the seeds. So the regeneration of the whole plant from the plant cells have been genetically modified. So the production of plants sterile containers reduce the disease transmission. It is very important, the disease transmission, and allows production of plants from the seeds that otherwise have very low chances of germinating and growing. So, for example, orchids and nepenthes. To clean particular plants of viral and those infections, to quickly multiply these plants, cleaner stock for the horticulture and agriculture purposes. So here, the terminology of in the tissue culture mostly we are frequently used in the some terminology in the plant tissue culture. That's explant. What is the meaning of explant? The living tissue transfer from a plant to an artificial medium for the culture. It can any portion of the shoot, leaves, root, flower, or cell from the plants. So, do you understand? Explant, living tissue transferred from a plant to an artificial medium for the culture. It can be any portion of the shoot, leaves, roots, flower, cells from the plant. This is the explant. So next, how? So the adult plant cells are totipotent. The meaning they have ability to give rise to a fully differentiated plant. What is the totipotence? Adult plant cells are totipotent capacity. That meaning they have the ability or capacity to give rise to a fully differentiated plant. Because of this, it is possible to collect the cells from the mature plant and use of these cells to produce the clones of the plants. So these are the root of carrot plant, root cells in the growing medium. It is the single cell division in culture, and it is the young plant, and finally we got the adult plant. So plant tissue culture basics. The modern plant tissue culture performed under the aseptic conditions. So here, living plant materials from the environment are naturally contaminated on their surfaces. Living plant materials from the environment are naturally contaminated on their surfaces, and sometimes with microorganisms, so surface sterilization and starting material, explants. into chemical solutions. Usually alcohol and sodium or calcium hypochlorite are required. So generally, the plant parts or surfaces to the contaminated with the microorganisms. So how to clean this uh, plant part surfaces? It is like explants. So it is the contamination. It is removed by the chemical solutions like alcohol, sodium or calcium hypochlorite is required. So here the explants then usually place on the surface of the solid culture medium, but are sometimes placed directly into a liquid medium when self-suspension cultures are desired. So the culture media are generally composed of the inorganic salts plus few organic nutrients and vitamins and plant hormones. These are the culture media. These requirements compulsory are necessary to develop a culture media. The culture media, it is composed inorganic salts, few organic nutrients, vitamins, and plant hormones. These are the requirement of the culture media. So next we'll go for plant tissue culture media. Salt mixtures, organic substances, natural complexes, inert supportive materials, and growth regulators. So we will discuss one by one. So the macronutrient salts, these are the all the macronutrients. What we call macronutrients, it is ammonium nitrate, NH4NO3, potassium nitrate, KNO3, calcium chloride, anhydrous, CaCl2 and 2H2O, magnesium sulfide, MgSO4, 7H2O. So next, potassium hypophosphate, it is KH2PO4. Next, ferrous and sodium ethylene diamine tetraacetate, FeNaEDTA. So H3BO3, it is the boric acid. MnSO4 and 4H2O. MnSO4 and 4H2O, it is manganese sulfate. ZnSO4, 7H2O, it is zinc sulfate. So next, potassium iodide, it is Ki. And next, sodium molybdate, Na2MO4 and 2H2O, sodium molybdate. Cupric sulfate, CuSO4 and 5H2O. And next one is cobaltous sulfide, it is CoCl2 and H2O. So next, some macronutrients, nitrogen, it influence the plant growth rate, essential in the plant nucleic acids, DNA, proteins, chlorophyll, acids, and hormones. And phosphorus, these are the what we call NPK. So abundant meristematic and fast-growing tissue, essential in the photosynthesis and respiration. So potassium, so necessary for the cell division, meristematic tissue, helps in the pathway of carbohydrates, protein, and chlorophyll synthesis. So the nitrogen is also very important. These three are the necessary of macronutrients for the tissue culture medium. Nitrogen, the plant growth rate, essential in the plant nucleic acid, proteins, chlorophyll, amino acids, and hormones. So phosphorus, meristematic and fast-growing tissue, essential in the photosynthesis and respiration, necessary for the cell division. Meristematic tissue helps in the pathway for the carbohydrate, protein, and chlorophyll synthesis. So coalition, it is also involved formation of cell walls and root and leaf development. Participates in the translocation of sugars, amino acids, and type of oxalic acid. So iron, it is involved in the respiration, chlorophyll synthesis, and photosynthesis. FeNaEDTA, sodium salt of EDTA, sequesters iron, making it available to the plants. The next one is the magnesium. It is the photosynthetic and respiration system, active in uptake of phosphate and translocation of phosphate in the starches. So next, micronutrients. So you know what is the difference between the macronutrients and micronutrients? Macronutrients, it is large, the large quantities are needed. The micro is very small quantities required to the plant growth. So it is the micronutrient sulfur. It is involved in the formation of nodules and chlorophyll synthesis, structural component of amino acids and enzymes. Manganese, involved in regulation of enzymes and growth hormones, assist in the photosynthesis and respiration. So here, next continuation of micronutrient salts. Molybdenum, it involved in the enzymatic reduction of the nitrate to ammonia, the assists to conversion of inorganic phosphate to organic form. Zinc, it is involved in the production of growth hormones and chlorophylls, active in the respiration and carbohydrate synthesis. The boron, the production of growth hormones and chlorophyll, active in respiration and carbohydrate synthesis. So copper, involved in the photosynthetic and respiration system. It is assists the chlorophyll synthesis and used as a reaction catalyst. So organic compounds.
Sir, page number, sir.
So page number is 273. So 73. So actually, this information, only little bit information is available in book. So I will elaborate in the other source. I will prepare these slide. So next, organic compounds, carbon sources, sucrose and sometimes glucose and fructose. And next one is vitamins. Adenine is a part of the RNA and DNA. Inositol, the part of B complex in the phosphate form, is part of the cell membranes, organelles, and is not essential to the growth, but is beneficial. Thiamine, the essential as a coenzyme in the citric acid cycle. So these are the organic compounds. Next, some and other organic acids. The citric acid, 150 milligrams per liter, typically used with the ascorbic acid. 100 grams per liter is an antioxidant. Can also some use Krebs cycle acids and phenolic compounds. Chloroglucinol, it is stimulates the rooting of the shoot section. Chloroglucinol. Next one is the natural complexes. Is a coconut endosperm, fish emulsion, protein hydrolysates, tomato juice, yeast extracts, malt extract, potato agar, and growth regulators. Auxins. Root auxins, it is used for the roots. Cytokinin, it is used for the shoots. Gibberellins, it is used for the cell enlargement. Abscisic acid, the plant stress hormone, the ethylene. And next one is axons. These are the axons, callus formation, root of cuttings, and the induction of advantageous embryogenesis. So it is IAA, indole acetic acid, IBA, indole butyric acid, NA, naphthalene acetic acid, 2,4-D, 2,4-dichlorophenoyl acetic acid, so 2,4,5-T, and picchloram. So cytokinins enhance the advantageous shoot formation. So carnitine and gibberellin, not generally used in the tissue culture. it is tends to suppress the root formation and advantageous embryo formation. So abscisic acid primarily as a growth inhibitor, but enables to more normal development of embryos, both zygotic and advantageous. So here ethylene the is not how much add but how to get rid the in vitro. The natural substances produced by tissue culture, fairly high levels, especially when cells under the stress. So enhances senescence and suppress embryogenesis and development in general. So callus development, advantageous embryogenesis, rooting of the shoot cutting, advantageous are the shoot and root formation. These are the hormone combination. These are the diagrams, schematic diagrams of the what we call this. It is the sterilization and implanting the explant for the culturing. Okay, these are the process. So the first is the plant, second is the surface. You see the book 275 mark. So surface 2 is the surface sterilized, slicing the explant. The next fourth is the aseptic plant, and seventh is the explant on cultured medium. So this is regarding about sterilization and implanting of the explant for culturing. So here the applications. The plant tissue culture is used widely in the plant sciences, the forestry, and horticulture applications. It include it is the commercial production of plants used as a potting, landscaping, or floristic subjects, which uses meristem and shoot culture to produce large number of identical individuals. To conserve the rare and endangered plant species, and a plant breeder may use the tissue culture to screen cells rather than the plants for the advantageous characters. Example, the herbicides, uh, herbicide resistance and tolerance. The large scale growth of plant cells in a liquid culture in bioreactors for the production of valuable compounds like plant-derived secondary metabolites and recombinant proteins used as a biopharmaceuticals. So, to cross distantly related species by the protoplast fusion and regeneration of the novel hybrid. And next, to rapidly study the molecular basis study the molecular basis for the physical and biochemical and reproductive mechanism in plants. For example, in vitro selection for the stress tolerant plants to cross pollinate the distantly related species, and then tissue culture resulting in the embryo, which would be normally die, it is embryo residue for the chromosome doubling and induction of the polyploid, for example, double haploids, tetraploids, and other forms of polyploids. This is usually achieved by application and anti-meiotic agents such as colchicine or origelin. So, as a tissue for transformation followed by either short-term testing of genetic constructs or regeneration of the transgenic plants. And the certain techniques such as meristemtic culture can be used to produce clean plant. The material from virus stock such as the sugarcane and potatoes and many species soft fruits production identical steroidal hybrid species can be obtained. And the large scale production of the artificial seeds through the somatic embryogenesis. These are the some basic plant tissue culture. So next we will go for these are the morphogenesis. So next we will discuss anther culture. So the page number is in your textbook in the Anthra culture is page number is 276 to 278.
Okay.
Yes, got it? 276.
Yes, got it.
Yes. So before going to Anthra culture and pollen culture, we discussed some terminology of on the haploid, the gametic number of chromosome and which may not be equivalent to X. So apply rapid regenerative homogenesis genotype after chromosome doubling, reduce time for variety of development. Just wait. Okay, in vitro methods. You know in vitro methods? Anther culture. It is also called androgenesis, production of haploid plant from the microspores. Anther culture of production of haploids reported about 250 species. For example, Solanaceae, Cruciferaceae, Graminae, Ranunculaceae, most common. And another one is the oval culture. So gynogenesis, what we called in the oval culture, gynogenesis, anther culture, androgenesis, the production of haploids, the plant from the unfertilized egg cells, it is applied. So next, the production of haploids in the in vitro through anther and microspore culture. So history. So Tulkey in the 1953 first observed that the mature pollen grains, Ginkgo biloba, as a gymnosperm member, can be induced to proliferate in a culture to form a haploid callus. So, and next, S. Guha and S. Panchanan Maheshwari in the 1964 first reported in the direct development of embryos from microspores of Datura inoxia by the culture excised the anther. So later J.P. Borgin and J.P. Nitsch in the 1967 obtained the complete haploid plantlets from the anther culture of Nicotiana tabacum. So anther culture. So anther culture is a technique by which the developing anthers at a precise and critical stage are excised aseptically from unopened flower bud and are cultured on a nutrient medium. Listen carefully. Anther culture is a technique by the developing anthers at the precise and critical stage are excised aseptically from unopened flower bud unopened flower bud and are cultured on a nutrient medium where the microspores within the cultured anther develop into a callus. Anther develop into callus tissue or embryoids that give rise to a applied plantlets either through organogenesis or embryogenesis. So androgenesis is the in vitro development of applied plants originating from the totipotent pollen grains through a series of cell division and its differentiation. It is two types, direct androgenesis, indirect androgenesis. The microspores behave like a zygote and undergoes a chance to form a embryoid which ultimately give rise to a plantlet. So indirect androgenesis, the microspores divide repeatedly to form a callus tissue, which differentiates into a haploid plantlets. These are the angiosperms reproduction. Already we discussed in the previous classes. So these are the sepals. Uh, these are the ovary. These are the style, stigma. The entire three processes it is called a carpel. These are the filament and anther. Filament and anther is called as the stamen. These are the petals. The principles of the anther and pollen cultures. The production of haploid plants exploding the totipens of microspore. In this process, a normal development and function of the pollen cell to become a male gamete is stopped and it is diverted the forcefully to a new metabolic pathway for the vegetative cell division. So, development of androgenic haploids pathway 1, the microspores divide by an equal division and identical daughter cells contribute to the saprophyte development. The vegetative and generative cells are not distinctly formed in this pathway. Example is Datura inoxia. So the division of uninucleate microspores is unequal, resulting in the formation of vegetative and generative cell. The saprophyte arises through the further divisions in the vegetative cell, while the generative cell does not divide. Example is Nicotiana tabacum. Pathway 3. The uninucleate microspores undergoes the normal unequal divisions. The pollen embryo are formed in the generative cells alone. Example is the Hyoscyamus niger. Pathway 4. The division of microspore is asymmetrical. Both vegetative and generative cells divide the further and contribute to the development of the sporophyte. Example is Atropa belladonna. These are the steps in the anther culture. Factors influencing the anther culture. The genotype of donor plants. The genotype of the donor plants plays the significant role in the determining the frequency of the pollen production. Example is Horodum of each genotype differs with the respect to androgenic response in the anther culture. Anther wall factor. The anther wall provide the nourishment in the development of isolated pollen in a number of species. There are reports that glutamine alone or combination with serine and myonosinol could replace the anther wall factor is isolated the culture. So culture media, factor in playing the anther culture media. The anther culture medium is requirements vary with genotype and probably the age of anthers as well as condition under which the donor plants are grown. Incorporation of activated charcoal into the medium has stimulated in the induction of androgenesis. The iron in the medium plays as a very important role in the induction of the haploids. The potato extracts and coconut milk and growth regulators like auxin and cytokinins are used for the anther and pollen culture. So, culture medium, 2 hormone groups without hormones, mostly dicots, most success with the Solanaceous species do not want to the anther wall to form the callus. With hormones, most non-Solanaceous species, many monocots require the hormones or complex organics such as coconut milk. The medium particularly important in cereals and rice to be able to produce green plants. A major difficulty was large number of albino plants that resulted. The sucrose ranges from 2% Dicotiana to 10% brassica. Anther stage. The microspore of pollen must shift from the gametic to sporophytic pattern of development. Most responsive cells for the haploid embryo formation are those between the tetrad stage of microgenesis to just past the first pollen mitosis. In most of the cases, anthers are more responsive when cultured at the unnucleate microspore stage. So wheat, barley, rice. Anther of some species gives the best response if pollen grain is cultured the first mitosis or later stage. That is, example is Datura and tobacco. Effect of the temperature. The temperature enhances the induction of the frequency of microspore androgenesis. The low temperature treatment to anther flower bud enhance the haploid formation. The low temperature affects the number of factors such as dissolution to microtubules, the lowering of the abscisic acid maintenance of higher ratio of variable pollen capable of embryogenesis. The physical status of donor plant. The physical status of donor plant, such as water stress, nitrogen requirement, and age of donor plant, highly affected the pollen embryogenesis. The plant starved of nitrogen may give more responsive anthers compared to those that are the well fed with nitrogenous fertilizers. Methods of anther and pollen culture. These are the diagrams. Cultures of isolated microspore. It is pollen. These are the flowering plant in the part. This is the inflorescence, the flower buds. 1, 2, 3, 4, 5, 6. Put the flower buds in the 5% of T-pol solution. Then wash in the running tap water. It is laminar airflow. So place the anthers on the culture media. So sucrose sterilization and wash in the sterile distilled water. So here dextrate the embryogenesis, it is the anther culture, is the indirect androgenesis. So initiation of callus tissue, these are the callus tissue. So organogenesis, indirect organogenesis. So regenerated the plantlet. So here same. So then we will regenerated applied plant in the pot. So here next process is here it is culture isolated from the microscope. It is pollen. So it is anther. Screen the anthers, filter the microspores, and again filter resuspension in the fresh medium, microspores, planting of the microscope. After embryogenesis, they are developing the regenerated the plantlets. So here, the advantage of pollen culture over the anther culture. During the anther culture, there is always possibility the somatic cells of the anther that are deployed will are also respond to the cultural condition and so produce unwanted diploid callier plantlets. Sometimes of the development of microspores inside the anther may be interrupted due to the growth inhibiting substances, the leaking out of the anther wall in contact with the nutrient medium. So advantage of the pollen culture of interest because formation of embryo is known to be from one cell only, thus no chimers are formed. Much more difficult than anther culture. Culture either isolated microspore or pollen, the Brassica oleracea. So next in the oval culture, this is the above the pollen and anther culture. So the oval culture also, I think the next page, just wait. The page number is ovary and oval culture is 279.
Yes, sir.
Yes, 279.
Okay.
So here, oval culture, the haploids can be induced from the ovals, right? So the number of ovals is less than that is used less than anther culture, maybe maybe by organogenesis or embryogenesis. So used in plant families that do not respond to androgenesis. The families is Liliaceae and Compositae. So The utility of anther and pollen culture for the basic research is the cytogenetic studies, study of genetic recombination in the higher plants, study of mode of differentiation from the single cell to whole organisms, study of factors controlling the pollen embryogenesis higher plants, the formation of double haploid that are homozygous and fertile. So anther and pollen culture are used for the mutation study. Example, nitrate reductase mutants are reported in the Nicotiana tabacum. Anther and pollen culture used for the plant breeding and crop improvement. Anther culture are used to obtain the alkaloid. Example is the homozygous recombination, high thymus, high niger having higher alkaloid content into obtained in the anther culture. Haploid are used in the molecular biology and genetic engineering. Example is haploid tissue of Arabidopsis and Lycopersicum have been used for the transfer and expression of 3 genes from the E. coli. So here, and another one is the embryo culture. So most important of the rescuing interspecific and intergeneric hybrids. Wide hybrids often suffer from the early spontaneous version, as cases embryo endosperm failure. So embryo culture, it is rescuing interspecific and intergeneric hybrids, Gossipum, Brassica, Linium, and Lilium, production of monoploids. The useful for obtaining haploids of barley, wheat, other cereals. The barley system use Hardium bulbosum as a pollen parent. Embryo culture, most the production of monoploids. The zygote develops into an embryo with elimination of HB chromosomes. Only HB chromosomes are left. The embryo is rescued by the culturing 10 pp to avoid the abortion. So here the hand pollination of freshly opened flowers, surface sterilization of ethyl alcohol and enclosing the structures, dissecting the scope necessary, plating on the solid medium, slain media are often used to avoid the condensation. The culture medium factors, the mineral salts like potassium, calcium, nitrogen is most important, and carbohydrate and osmotic pressure. 2% of sucrose works well for the mature embryos. 8 to 12% of the immature embryos. Transfer of progressively lower levels as the embryo goes. High sucrose, axinin, cytokinins. The culture medium factors, amino acids. Reduced nitrogen often the helpful. Up to 10 amino acids can be added to replace the nitrogen salts. It includes glutamine, alanine, arginine, aspartic acid. So it requires the filter sterilizing a portion of medium. So the culture medium factors, natural plant extracts, coconut milk, it is the liquid endosperm of coconut, enhanced the growth attributes through undefined the hormonal factors and organic compounds. Others, the extracts of dates, bananas, milks, tomato juice. So this one is globular embryos require the low concentration of oxygen and cytokinins, hot stage and later non-required. So gibberellic acid and abscisic acid regulate the precious germination. Culture medium factors, gibberellic acid and abscisic acid regulate the precocious germination. Gibberellic acid and ABI suppresses. So in vitro pollination and fertilization. These are the in vitro ovular pollination. A flower bud is cultured in a nutrient medium. Aseptically collected pollen is applied directly to exposed ovules in vitro. So intergeneric hybrids, Caryophyllaceae, interspecific hybrid is the Solanaceae and Brassicaceae. So emasculate and cover the flower buds to control the pollination and collection of the pollen grains. Remove the sepals and petals. surface disinfected, except pistil 70% ethyl alcohol, rinse with the sterile distilled water, place the pistil into the culture. So the pollination through a slit or pore, pollinate on the stigma, cut up the pistil into small pieces of the placental tissue with the attached ovules, culture individual ovules, the collecting pollen, surface sterilized buds with anthers, keep the sterile petri dishes till anthesis. The anthers are taken from the open flowers and pollen is collected and applied to culture ovules, placenta or stigma depend on the methods. So factors affecting the seed set after the pollination, the less parental tissue removed, better seed set is the latter. Some species like mice are more tolerant than others. So not wetting in the surface of the ovules or stigma, time of exercising of the explant. So a pollinated pistil provides better ovules that later have a better seed set. The medium requirements, simple minerals, salts, and few vitamins and sucrose. The sucrose at 4 to 5% is typical, but some workers use higher levels. So the pollinated pistil provides better ovals that later have better seed set. Medium requires simple mineral salts, a few vitamins, and sucrose. The sucrose at 4 to 5% are typical, but some workers use higher levels. So some have used simpler technique than any presented here. The culture of ovules after pollination in vivo. So Gossypium, Arboreum, Hirsutum, and Trifolium repens versus Hybridium, Helianthus annuus and Maximiliani, Helianthus annuus and tuberosum. So these are the true in vitro fertilization. Only GMS using the single egg and sperm cells and fusing them electrically. Fusion products were cultured individually in the mill cell inside the layer of feeder cells. The resulting in the embryo was cultured to produce a fertile plant. Unsuggested, the fusion genetically is modified gametes. So these are the regarding about anther culture and pollen culture and ovule culture. So these are the different cases. We'll see, just wait. These are the callus culture. Different cultures like suspensal culture, so continuous culture, batch culture. Suspension culture is a type of culture, is a single cell protoplast or small cell aggregate multiple the agitated in the liquid medium. So here in the batch culture, continuous culture, the regeneration of the plants through the cell suspension culture and protoplast culture. So next, meristem culture. This is the shoot tip. You take the shoot tip, surface of sterilization by sodium hypochlorite, and then distilled water, then scale peel. Apical tip, it is Murashi and scoop medium. So that means here all the necessary organic and inorganic hormones and as well as vitamin also in this MS medium. MS medium is Murashigi and scoop medium. To incubate the culture for the 16 hours at the 25 degrees Celsius under the fluorescent light, it is automatically the development of the root, then transfer to the pods. It is the meristem culture. Next, root culture. So these are the root culture, regeneration of plants from the root exploring the liquid medium. It is applied culture. These are the production of applied plants using the anther tissue. So we already in the previous discussion, this is the flower. So you take the anther here, then anther culture in the nutrient medium. So embryonic here applied plantlet, and it is the callus and applied plantlet. So next, anther and pollen culture. What is the anther culture is technique by which immature anther is made into divide grow into tissue, either callus or somatic embryo, primarily to produce haploid plants with the number of chromosomes, haploid set of chromosomes. The pollen culture, microspore culture, is a technique which haploid plants are obtained from the isolated pollen grains. This one is the pollen grains. That's one is the anther culture. So here the are pollen grains are dusted on a suitable culture medium and allowed to grow into the callus somatic embryo. Plants can be regenerated through the shoot and root induction in the applied callus through the culture of haploid embryos. It possible to produce homozygous double haploid pure breeding lines through the chromosome doubling treatments applied the haploid cells. And Guha and Maheshwari were first produced the plants Datura inoxina through the culture anthers containing the immature pollen. This technique was discovered by Guha and Maheshwari in the 1964. This one. Animation, the culture, solid media, this the selecting the anthers, remove the filament, the culture in the liquid media. So the production of the, and this is the another technique, the ovary and oval culture. The oval culture is an elegant experimental system in by which ovals are aseptically isolated from the ovary and grown in the aseptically and the chemical defined nutrient medium under the controlled conditions. The ovary culture is the technique of culture of ovaries isolated either from the pollinated or unpollinated flowers. When the tissue pollinated and fertilized ovary is taken, they produce the diploid plants. When the unfertilized ovary tissues are taken after it has gone meiosis, it will result in the formation of applied plants. There is a scarcity of applied cells within the oval as a compared to the anther. So anther are microscope culture generally used to the develop applied plants. The new first reported successfully oval culture barley. It is helpful in achieving the in vitro fertilization. It can be used in the recovery of hybrid embryos, which gets aborted with the incompatibility of the parent. This is the diagram of the ovary culture. So another one is the embryo culture. The embryo culture is the culture. It is isolated, immature and mature embryos. The zygotic or seed embryos are often used in the advantageously as an example in plant tissue culture. For example, initiate of the callus culture. This embryo develops properly when nourishing the tissue. The endosperm remains present in the seed during the development. The embryo culture is applied to overcome embryo abortion due to the incompatibility barrier, embryo rescue to overcome seed dormancy and shortening the breeding cycle. So these are the procedure of embryo culture. So these are the cut point immature embryo. The explant embryo is collected from the surface of the sterilization. It is the next the embryo in the capsula seed. It is the embryo. It is like in barley seed. It is embryo put on a culture medium. It is next to step is callus formation, organogenesis, then formation of the plantlet development. Then here the several embryogenesis that are differentiated to embryo and seedling of medium. So these are the somatic embryogenesis. These are the diagram. So to explant and the nutrient media, then callus, embryo, and explant. So regeneration of plants through the somatic embryo is the father of plant tissue culture. Haberland. Okay. So contribute the Armenian scientist toward the plant tissue culture. E.C. Cooking and is the white Novport, Perrier Roger, and these are the scientists. So these are the today class. Today we will discuss experimental embryology and plant tissue culture and preparation of MS medium and pollen anther culture, pollen culture, embryo culture. So some other type how there are steps going on. So any the plant tissue culture in this The plant tissue culture, the collection of techniques used maintained. So these are some importance of what are the importance of plant tissue culture. So plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues, organs. So the plant tissue culture is used as a collective term to describe all kinds of the in vitro cultures. So these cultures can be cell culture, a tissue culture, or an organ culture. So cultures are generally initiated from the sterile pieces of the plants known as explants. What is the meaning of explants? Cultures are generally initiated from the sterile pieces of plants known as explants. Explants may comprise an organ tissue like it is shoot tip, node, leaf, root, or stem, a cell, protoplast, pollen, ovum, and endosperm. The plant tissue culture is widely used by the plant science, forestry, and horticulture. It is used in the first one is the clonal propagation and micro propagation. The second one is in production of virus-free plants. Third one is in germplasm conservation. Next, fourth one is to produce sterile plants. In production of secondary metabolites. In making of artificial seeds and synthetic seeds. In production of transgenic plants. In production of hybrids and hybrids. In production of haploids, triploids, and dihaploids, and in production of polyploid plants. So these are the uses of 10 uses. The plant tissue culture, it is used by in the plant science, forestry, and horticulture. So here, so clonal propagation, micro propagation, production of virus-free plants, and the germplasm conservation. These are the very, very important things. That's why I'm repeating the second time. Germplasm conservation through the tissue culture. To produce sterile plants through the tissue culture. And production of secondary metabolites through the plant tissue culture. In making artificial seeds or synthetic seeds through the plant tissue culture. In production of transgenic plants through the plant tissue culture. The production of hybrids and hybrids through the plant tissue culture. In production of haploids, triploids, and dihaploids through the tissue culture. In production of polyploid plants through the tissue culture. These are the 10 types of uses through the tissue culture. So this, the first of all, in the tissue culture step, in the first initiation step is the callus. The callus means it is a undifferentiated and unorganized mass of parenchymatous tissue. It is called as callus. It is very, very important. In this tissue culture, the formation of callus is very, very important. In every step, most of the steps, it is compulsory, the formation of callus. So we will get the some plants and plantlets through the callus culture also. So first of all, callus. What is the callus? The plural callus are calli. It is a undifferentiated and unorganized mass of parenchyma tissue called as callus, undifferentiated and unorganized of parenchyma tissues. So in living plants, the calluses are undifferentiated mass of cells which cover a plant. In biological research and biotechnology, callus formation is induced in explants after the surface sterilization and inoculation of explants and optimal aseptic media in vitro condition. The culture medium is supplemented with an identified amount of plant growth regulators such as axins, cytokinins, and gibberellins to initiate the callus formation of somatic embryos. The formation of initiate the callus formation or formation of somatic embryos. During this process, cell differentiation and specialization is reserved and explant gives rise to the new tissue, right? During this callus culture, what happening here? During this process, the cell differentiation and specialization is reversed and the explant gives rise to the new tissue. The callus, which is formed and an original explant, is called primary callus, which is compact. The secondary callus are formed by subculturing the primary callus. The repeated subculturing forms preable callus. So these are the you observe this diagram. Preable callus and compact callus. And another one is the suspension culture. So already I discussed it. So here in this important meristematic culture also very, very important. So meristem culture. So here the meristem culture is meristem tissue shoot tip of in vitro in a suitable culture medium. This type culture is done mostly of the elimination of the viruses and related pathogens from a large number of vegetatively propagated plants. It is the main method used in the plant virus elimination programs. It is used for the mass production of desirable genotypes and helpful of the in clonal propagation. This is the meristem culture. So first of all, you cut it in the shoot tip. Then this shoot tip, it is surface sterilization by the sodium hypochlorite chemical. Then it is transferred to the distilled water. Then scale the peel. This peel, it is will get the apical tip. Then it is transferred to the MS medium. Already I told you. So MS medium is the Murashigand's food. It is related to the it is required this already in the startingly we discussed. In this medium, the preparation of in this MS medium, how many types? The organic salts, inorganic salts, and vitamins, plant hormones, micronutrients, macronutrients. These are the combination of some combination some of the chemicals which is required in the little bit. So it is already a structured program. So, and the preparation of the MS medium, the apical tip, dip on the MS medium, put on the MS medium, then incubate the culture for the 16 hours at the 25 degrees Celsius under the fluorescent microscope. Then what happening here? The root development is initiation of root development. So then this plant, after development of root, this plant transferred to the parts. After transfer of the parts, then it is transferred to the fields. So this is the meristem culture. It is the very, very important. So the production of applied plants using the anther tissue, already we discussed. This is the flowers. So that's why I many times I told you. First of all, what are the reproductive structure of reproductive parts of the flower? You identify, then you will easily get the understand anther. So this is anther culture. First of all, in this, you take the anther, and the anther put in the anther culture nutrient medium. Then it is automatically, it is what happening in this applied plantlet. It is the anther culture. Through the anther culture, what we will get? It is the applied plantlets will get. So it is the formation of colors and applied plantlet. This is the using the anther tissue. So this is also regarding. So already I told you different types of the anther cultures. So one most important, through the tissue culture we produce in the secondary metabolites. Why do plants produce secondary metabolites? So these are the secondary metabolites in the plant cultures, applications, and production. The production process comprises the seven aspects. The seven aspects is the selection of the cell lines for high yield of secondary metabolites. This is the first one. Second one, the large scale cultivation of the plant cells. Third one is the medium composition and effect of nutrients. And elicited induced production of secondary metabolites. Effect of environment factors. Sixth one is biotransformation using the plant cell cultures and secondary metabolites release and analysis. The secondary metabolites, the chemical compounds produced by plants are collectively referred as the phytochemicals. The chemical compounds produced by the plants are collectively referred as the phytochemicals. The biotechnologists have special interest in the plant tissue culture for the large scale production of commercially important compounds. These includes pharmaceuticals, flavors, fragrances, cosmetics, food additives, and feedstocks and antimicrobials. Most of these products are secondary metabolites, chemical compounds that do not participate in the metabolism of plants. The secondary metabolites are not directly needed by plants as they do not perform any physiological function, as the case with primary metabolites such as amino acids, nucleic acid. Although have native plants are capable production of secondary metabolites, commercially interest tissue culture system are favored. What are the advantage and limitations are lifted? These are the major advantages. Compounds can be produced under the controlled conditions as per the market demands. Culture system are dependent on the environment factors, seasonal variations, pest and microbial diseases, and geographical constraints. Cell growth can be controlled to facilitate improve the product formation. The quality of the product will be constant. It is produced by the specific cell line. The recovery of the product will be easy. Plant cultures are particularly useful in case of the plant which are difficult to expensive to grown in the fields. The mutant cell lines can be developed for the production of novel compounds of commercial importance which are not normally found in the plants. Biotransformation reactions. The production of control is not mercy of political interference. The production of time is less labor cost in the minimal. So these are the some advantages. Disadvantages: the in vitro production of secondary metabolites is lower when compared to the intact plants. Many times secondary metabolites are found in the differentiated the tissue organs. In such case, culture cells which are non-differentiated can produce the little. The cultured cells are genetically unstable and may undergo mutation. The production of secondary metals may be drastically reduced in the culture ages. Vigorous stirring of necessary to prevent aggregation of culture cells may obtain damage to the cells. These are the some disadvantages of secondary metabolites through the tissue culture. Based on the existing evidence, it is believed to the production of some secondary metabolites linked to the induction of the morphological differentiation. Consider the cardiac glycosides are found in the leaves of digitalis. Quinine and guanine are present in the bark of cinchona. Tropane alkalides are found in the roots of the atropa. It appears that cells undergo morphological differentiation in the maturation during the plant growth. Some of the cells specialized to the produce metabolites. It is also observed that the in vitro production of secondary metabolites is much higher from the differential in the tissue when compared to the non-differential or less differential tissues. These are the regarding about through secondary metabolites through the tissue culture. So this is the today class. Thank you.
Okay, thank you.